Hi, my name is Daksha and my metric number is 185091, and today I'm going to talk to you about the enzyme kinetics lab that we had. First, let me define what enzymes are. Enzymes are generally protein structures that are used to speed up a certain reaction, and they are not used by the end of the reaction, as in they can be recycled. The reactions generally are for the breaking down of certain molecules, and also to build up certain molecules, and each type of enzyme has a specific shape that enables the substrate to bind to. That's also called the active site. There are also other molecules that could bind to the enzyme that could activate or deactivate it. These could be at a site other than the active site, or it could be at the active site itself. The shape and the chemical properties of the active site is very specific, and it directly correlates to the enzyme's function. There are many chemical reaction that occurs generally at a much slower rate when enzyme is not present. Therefore, the presence of an enzyme can speed up the reaction, making the final product faster. The final product can then feed back on the enzyme, either to shut it down, to deactivate it, or to slow down the reaction. So in the enzyme kinetics lab, what we used was we would put in enzymes into conical flasks or conical tubes, and we would measure the activity of the enzyme over a period of time. And we measure this activity based on using a spectrophotometry based on the coloration of the conical tubes, the coloration of the solution in the conical tubes. So the brighter the brighter yellow colored conical tube means more reaction has taken place, and the less yellow colored tube would mean less reactions have taken place. We also used temperature as a manipulative variable for this experiment. We used solutions containing different temperatures, and we would incubate them with the enzymes to see how well the enzymes function. In this case, our enzymes were able to function up to 90 degrees, because cellobiose could sometimes be considered as a bacterial enzyme that could function under very high temperatures. Besides that, there is also a lot of preventative measures that we need to take so that we do not alter our results. One of it would be to handle the conical tubes with care, and to always wear gloves when handling the conical tubes. We should not place our fingers at the bottom part of the tube, which is going to go into the spectrophotometer, because that's the part that's going to have light reflected into it. Besides that, the timer should be set precisely. Everything should be measured accurately. Measurements should be taken once the device is already tailored, to prevent any errors in the machine. However, human error is possible, especially when we're using the timer and we're putting in the solutions. There could be a variation of time by a couple seconds, which could potentially alter your results. So that's what we did in the enzyme kinetics lab, and that's it. Thank you.